as you work through your AS biology course, you're going to find more and more places where topics are linked together. And it's really important that as you start a topic and as you start a new topic, you're always looking for those links. It helps to consolidate your knowledge and it helps to make things make more sense. Today, we're going to look at one of those special links, linking the mitochondria, ATP, and respiration. So if we think about these three, they are very tightly tied together. Okay, if we're going to start this lesson with the mitochondria, we're then going to look at aerobic respiration. And we're going to finish up by considering ATP. So what you've got here is the site of respiration, the function that is happening with the respiration, and the output of respiration. So none of these could actually exist as a single thing. They're all so tightly linked together because effectively it is one thing that's happening, production of ATP through aerobic respiration in the mitochondria. So at AS, mitochondria and ATP definitely are on your radar. Aerobic respiration is actually covered in far more detail in the A-level portion of the course. But we're going to touch on it today just so that you've got an idea of how everything fits together. So starting with that all-important location, the mitochondria. So this is one of the organelles in your cell. We're talking about approximately 1 micrometer in diameter and approximately 10 micrometers in length. But we do get some variation within that. So diameter and length, we are talking of a sausage-shaped organelle. So if we take a transverse section, we have something that looks roughly like this, with all these folds on the inside. And as usual, structure and function are very tightly linked. So we'll get the structure going and then we'll have a look at everything that's going on. This pen doesn't like doing dots so much, so you can imagine that these are beautiful dots. Okay, so what have we got? Let's start with the outside, the membranes. Okay, so we have an outer membrane. We have an inner membrane. And we have what we can call an intermembrane space. So the space between the two membranes. Because we've got two membranes, we can call this a double membrane, also known as an envelope. Okay, so we have a double membrane on the outside of this mitochondria. And you see the outside one is just smooth and simple and goes all around the outside. The inside one is folded. And these folds are critically important, so they have a special name as well. One is called a crista. So plural, cristae, just add an E on the end. And what this is, it's an infolding of that inner membrane, and when you fold the membrane, you increase surface area. And of course, if we're increasing surface area, there must be some function that is linked to the area of that inner membrane. So this is the membrane that's now on the outside of our mitochondrion. What's going on further in? Well, the inside of the mitochondrion, we call that the matrix. So sort of the equivalent of the cytoplasm of the mitochondrion. Okay, but it's not cytoplasm because the mitochondrion is not a cell. So we call it the matrix. Then we have another couple of very important features. This wobbly loopy thing here is a loop of circular DNA. So DNA like you find in a prokaryote. Okay, no proteins, no histones, nothing like that going on, just plain old DNA. And then these attempted dots are 70S ribosomes. Again, like you find in prokaryotes, the small ribosomes. Smaller place, smaller ribosomes. So our circular DNA and our 70S ribosomes, DNA and ribosomes, all about making proteins. So these guys together, we're talking about protein making machinery. For the mitochondria. So some proteins for the mitochondria actually come from the cell itself, encoded with their instructions in the DNA of the cell. But the mitochondria can make its own proteins that it needs for its specific purposes as well. Another cool thing about the mitochondria is they can replicate. They can move, they can replicate. They can grow in size. Okay, so if you think about it, when a cell is dividing, they're going to need more mitochondria for the two daughter cells. So we have to be able to make more mitochondria, and these guys can replicate. So we can replicate with 1 by 10 micrometers sausage-shaped organelle with a double membrane system that in the membrane is folded, increasing the surface area. The matrix is just the inside space of the mitochondria. And then we've got the circular DNA and the 70S ribosomes contributing to that whole idea of protein making machinery, because everything, remember, is controlled by enzymes, and that includes aerobic respiration. So if we're comfortable with the site of where this is all happening, let us talk about aerobic respiration itself. Aerobic respiration. So again, a reminder, you don't have to know this in full expanded detail. That is covered in the A-level portion of the biology syllabus. For AS, we need to understand the basics, and we need to understand how it fits in and why it's happening in the mitochondria. Okay, and it is all about that inner membrane folding, those cristae. And what happens there is we actually have electrons that move through those cristae, and that creates energy. So electron movements through the cristae create energy. So now you see why it's really important to have that increased surface area forming the cristae, because the more surface area, the more electron movements, the more energy. But we can't just have energy zapping out of our cristae. We've got to lock it up. We've got to keep it safe. So that is what ATP is there for. That energy that's created by these electron movements is locked into ATP. Where does that happen? In the matrix. Okay, and it's all enzyme controlled. So energy is formed by the movement of these electrons locked into ATP. Why ATP? ATP is a fantastic energy currency, okay, which means that we can constantly recycle it, we can use it to give energy, then we can put energy back into it. So the same like we use money, okay, we can pay for something, then we get money back from earning it, and then we pay it, and it's constantly going around, okay, so that's the same thing that happens with ATP. So in the mitochondria, we have these cristae. In the cristae, we have electron movements. The electron movements create energy, which is locked into ATP. And yet these are all found in different chapters of your book. So you're starting to see why it's important to make these links between chapters, between topics, to solidify things. So to really understand how ATP is sort of forming in all of this and how we're sort of talking about this recycling of ATP, we need to look at ATP in more detail. Let us consider ATP. Full name, adenosine triphosphate. So it's our energy currency, and we've said it's constantly recycled. So let's have a look at that in a bit more detail. What it means is that we have ATP. That ATP goes to ADP, from tri to di, so 2 phosphates. Well, if we just add another phosphate group, then we would be able to make ATP again, and we can start all over again. So ATP to ADP, this is because of use in the cell. We add in P subscript I, that is what we generally use to indicate a phosphate group. And this is catalyzed, this ADP plus PI, catalyzed by enzymes as part of that aerobic respiration. So we're constantly losing a phosphate, gaining a phosphate, losing a phosphate, gaining a phosphate. So it's all about that phosphate. So if we think then it might be useful to actually have a look at what ATP looks like. So our structure of ATP is very much like the nucleotides we find making DNA. So that would be our nucleotide for DNA, but we don't just have one phosphate for an ATP, a triphosphate, we actually have 3 phosphates. So we have ribose, which is our pentose sugar. As the name suggests, our nitrogenous base is adenine. And triphosphate, we have 3 phosphate groups. So what happens is that we're going to lose this last phosphate group, and we're going to make ADP. Then we're going to do aerobic respiration. We're going to put that 3rd phosphate group on again. Then it's going to go and it's going to get used so that the energy can be used somewhere in the cell. And then we're going to put it back on again. So it's a constant recycling between ADP and ATP. What's really cool about ATP is that it is small, it is soluble, it moves easily around the cell, it moves rapidly around the cell. So even though it's being made in the mitochondria, it can be used anywhere very quickly. So it's a really good currency for energy, because we can't just have energy randomly zapping all over our cell. We'd run into all sorts of problems if that was what was going on. We'd really have to control that energy movement in the cell. And ATP is a fantastic way to lock up that energy to transport it to where the energy is needed for particular reactions. So we keep saying energy is needed for particular reactions. How? What happens? We've referred to it a little bit in terms of we can lose this phosphate group, but there's actually 2 different options here that works really nicely. So ATP provides energy for reactions. And there is two different ways in which this can be done. As we've talked about, we can actually hydrolyze that last phosphate group and that releases energy. So we would call that in fancy terms, hydrolysis of terminal phosphate group. So that then releases the energy that can be used in order to have the reaction happen. The other option is that ATP could actually react with the metabolites, the things that are involved in the reaction, in order to make them phosphorylated. Now, when you phosphorylate something, you increase its reactivity. So it's the same effect as giving energy to the reaction because you make the things more reactive. Let's actually start with the different, let's say ATP. So we can either directly donate energy through that hydrolysis, or we can create phosphorylated intermediates, which are more reactive. So everything from the structure of a mitochondrion through aerobic respiration to ATP. And yet these things are found in different places, but they're also tightly linked. So use this as an example and go hunt for other linkages. Go hunt for other networks you can find between topics, because it really does make things a lot easier when you've got them in context and you can see how they relate to each other. Also, of course, make sure that you are happy with the structure of the mitochondria and how that structure is linked into the function of the mitochondria, aerobic respiration, and the production of ATP.